Hello class, I'm Jose Diaz, and today I'm going to be presenting what we've done so far in quarter 3 in our biology class. So to begin with, also known as Meiosis 1 and Meiosis 2, PMAT 1 and PMAT 2 are used for just one purpose in the human body, and that is the production of gametes, sex cells, or sperm and eggs. Its goal is to make daughter cells with exactly half as many chromosomes as the starting cell. Something important to know is that humans have 46 chromosomes. These are split evenly, meaning that 23 chromosomes come from each parent. These also known as sex gametes. So, meiosis 1 is a process divided into 4 phases, which are the following: prophase 1, metaphase 1, anaphase 1, and telophase 1. Now, I will move you guys to my iPad where I will be explaining and modeling this process. So, we first start with this germ cell. As you can see, there we have our cell membrane and the nucleus. Cell membrane, nucleus. Due to spacing, we will be working with 4 chromosomes that replicated due to interphase. Though, as I said before, humans have 46. These are split evenly, meaning that 23 chromosomes come from each parent. We have a homogeneous pair to the left and another one to the right. So, we're moving into prophase. Prophase is the first phase of meiosis, a process that separates the duplicated genetic material carried in the nucleus of a parent cell into 2 identical daughter cells. Also, the nucleus disappears. During the prophase, the complex of DNA and proteins contained in the nucleus, known as chromatin, condenses. As you can see in pink, right here, the chromosomes are being held together, forming tetrads. So crossing over happens. You can see it right here. Now we're moving into metaphase. In metaphase 1, the tetrads line themselves up at the metaphase plate and homologous pairs orient themselves randomly. Look, here is them lining up. Now we move into anaphase. In anaphase 1, the homologous are pulled apart and move apart to opposite ends of the cell, as you can see right here. Pulled apart, moving to opposite ends. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart, as you can see right here. The pink is representing their attachment. Finally, now we're moving into telophase 1. During telophase 1, the chromosomes are enclosed in nuclei. You can see here a nucleus has formed. The cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into 2 daughter cells. Here you can see it's divided. The cell has been divided in 2. Each daughter cell is haploid and has only 1 set of chromosomes, or half the total number of chromosomes of the original cell. So we're moving into meiosis 2 now, which is like meiosis 1 that has 4 phases. These are prophase 2, metaphase 2, anaphase 2, and telophase 2. So in prophase 2, the nuclei and nuclear membrane disappear. As you see here, no more nucleus. The chromatids become short and thick. The centromeres move to the polar regions and arrange spindle fibers for the second meiotic division. In metaphase 2, so not much really happens. They just lose this nucleus, as you can see. In metaphase 2, it's the second process of meiosis 2. During this phase, the sister chromatids are maximally condensed and aligned at the equator of the cell, as you can see here. Now, in anaphase 2, the sister chromatids separate and are pulled towards opposite poles of the cell. See, no longer in the equator and no longer together. And finally, in telophase 2, nuclear membranes form around each set of chromosomes and the chromosomes decondense. Look, here you have they each got separated and got nucleus. Apart from all of that, we have learned a lot of new vocabulary words. I've used a lot of them in the video, but these are the ones I didn't use. So, the first one was a gene. A gene is the basic physical and functional unit of heredity. Genes are made up of DNA. Some genes act as instructions to make molecules called proteins. An allele. An allele is a variant form of a gene. Some genes have a variety of different forms which are located at the same position or genetic locus on a chromosome. Humans are called diploid organisms because they have two alleles at each genetic locus, with one allele inherited from each parent. A trait. A trait is a specific characteristic of an organism. Traits can be determined by genes or the environment, or more commonly by interactions between them. And gonads. The gonads are the primary reproductive organs, are the testes in the male and the ovaries in the female. Haploid cells. Haploid describes a cell that contains a single set of chromosomes. The term haploid can also refer to the number of chromosomes in egg or sperm cells, which are also called gametes. Fertilization. The process of combining the male gamete or sperm with a female gamete or ovum. The product of fertilization is a cell called a zygote. And finally, the diploid cell. A diploid describes a cell that contains two copies of each chromosome. Nearly all the cells in the human body carry two homologous, or similar copies of each chromosome. Thank you guys for watching, and I hope you enjoyed.